Hello, everyone, and welcome to the Cancer Genomics Cloud webinar, October 2022. It's my pleasure today to introduce Dr. Min Zhang from Purdue University. She's a professor at the Department of Statistics and the Associate Director of Data Science at Purdue University for the Center of Cancer Research. She obtained her medical degree and residency in cancer and PhD in neuroscience. And she received another PhD from the Department of Biological Statistics and Computational Biology at Cornell University. She's incorporating biological knowledge in statistical modeling, and she has focused on developing new statistical methods to analyze high-dimensional clinical and omics data that often occurs in biomedical research. Today, she's going to present on how to do multi-omics analysis in the Cancer Genomics Cloud, as well as her experience in using the CGC as a training resource in one of our classes. And with that, I will just take it away. You can share your screen.
Okay. Okay, can you see it okay now?
It's perfect.
Okay. Yeah, thank you for the nice introduction and also the opportunity to share my experience and also some of our collaboration work with the CGC team. Okay, so my journey in cancer actually started from middle school when I lost my mom to cancer and decided to go to medical school. So I did my residency in cancer, and also after maybe a decade in medical school, I realized the data is very important and switched my career to statistics. And currently I'm trying to bring my statistical and computational expertise back into the medical world. So there are two parts of my talk, and for the first talk, I'm going to focus on sharing my experience of the CGC workshop. So this is a co-organized with a group from Seven Bridges, and here is the outline of my talk. So the CGC workshop was initially motivated by the big data workshop that my colleagues and I are running. So with Dr. Nadia Lama and Dr. Zhang. So this is a project funded by NCI through R25 mechanism, and it's designed specifically for cancer researchers. So during the workshop, we provide the computing resources and teach the participants on how to analyze different types of omics data. And at the end of the workshop, there were always questions from the participants who don't have access to HPC, and they always ask about how can they keep, analyzing their own data after the workshop. And to solve this issue, we invited people from Seven Bridges and talked about the CGC platform. And the presentation was very well received all the time for all years. And since our big data workshop can only recruit 30 people each year, we have been thinking about how to reach to more cancer researchers. So after discussing with people from CGC and also the bioinformatics core at Purdue Cancer Center, so we decided to organize a series of lectures as part of the weekly bioinformatics seminar at our department. So before the workshop, we were trying to determine which topics to cover, since there are so many different type of analysis, currently in the big data world. So we sent out a survey to the to graduate students and also members of Purdue Cancer Center and some other faculty on campus, and try to get an idea what are they most interested in. So you can see from the pie chart, among the 5 categories, the bulk RNA-seq and the single cell RNA-seq occupy almost 60% of the people who are interested in learning about. So one of my PhD students, Danny Liu, conducted a survey for the pre-workshop, and we did another one after the workshop. So based on these results, we focus on the bulk RNA-seq and the single-cell RNA-seq in the workshop. So there are 4 lectures in total. So the first lecture is overall introduction to the CGC. So Dr. Zia Woolman introduced the CGC platform, and she also highlighted a couple of features on importing the data and running the analysis. And toward the end of the lecture, she asked everybody, log in and set up their own project. And here's the URL for the recording of the lecture. So we recorded all the lectures and also post the slides online if you're interested. So in the second lecture, Dr. Phil Webster started to talk about the bulk RNA-seq data analysis. So he started with an overview and the workflow editor, and then described the tools and steps to create a workflow. And he also did a hands-on demo for the CGC RNA-seq workflow. So in this one, we used a case study. So these slides and the next one is a courtesy from Dr. Ron Beck, that with nice pictures. So this is the data from Dr. Andresani's lab. So essentially, her lab has been focusing on liver cancer. So in this study, so they used the HepAD38 cell lines and to look at the DDX5 gene, the difference between the wild type and the knockdown. So if you want to look at the details, so the paper was published in Theranostics back in 2020. And the nice thing about this project is so the data is very well organized and is publicly available through the GEO website with a session number GSE 131257. So as a CGC platform, there is also specific tool to help importing the data from, and also the associated metadata. So if you have any data that you're interested in from either GEO or some other databases, so the tools are ready there that you can just use the tool to import data to the CGC platform. And in the next lecture, so Dr. Webster kind of dig down into the details of the bulk RNA-seq analysis. So in terms of how to do the differential analysis and how to visualize the results, the data, and also some post-alignment QC. Again, these are courtesy slides from Ron. And so we can see that, based on the 2 populations, and some of them, like on the right-hand side, shows the difference in terms of expression for the DDX5, and the bottom one indicates if there's no difference. So that's how we can actually use the CGC platform to import the data to do the analysis and visualize the results. And then we did the same thing for the single-cell RNA-seq analysis. So Dr. Manisha Rui gave the lecture on the single-cell RNA-seq data analysis. So she first started with why do we worry about the single-cell analysis, and show the difference between the bulk RNA-seq and the single-cell RNA-seq data. So the tools are available for the single-cell RNA-seq data analysis. She did a demo, and the recording of the lecture is also posted online. So the data we use is from Dr. Radcliffe, who used to be the director of our cancer center. So his research is more on prostate cancer, and this is a particular one related with a carcinoma-associated fibroblast. And the bottom, like the picture, shows the gene expression of the single cells of the 3 different genes. And again, the data is also available at GEO with a session number GSE 123359. So at the end of the workshop, we did another survey, just try to see how well we did or whether we meet the expectations. So this figure shows the difference, like, on the left panel, so the pie chart indicates only 18% of the participants use the CGC platform before, but after the workshop, combine the already used and plan to use, so it's almost 80%. And I did talk with some of the participants. They said, "Oh, we even didn't know the existence of that, and we're really excited to use this for our own research." And in terms of the overall expectation, so we give 4 options, going from poor, fair, good, excellent. And almost all of them, have selected the good and excellent. So here are some open-ended comments that we collected on the post-course survey. So this is just a short list of them. So you can see most of them, they learned how to use the framework in the CGC platform. And as most of our participants are cancer researchers, so they feel that it's very efficient and easy for them to use this, especially they don't have much computing background or coding experience to do the data analysis, which used to be really scary with high dimensional data. And also some of them, if they don't have their own data, but they want to use CGC as a resource to access data and analyze them directly, and they found it quite convenient to find the data that they're interested in and analyze directly with the CGC platform. And one of the PI even told me specifically that her students is currently working with CGC technicians on some specific projects or questions they are trying to ask, using the public database already. And not surprisingly, after the workshop, a lot of them asked me about, when will the next series be happening? Because after the bulk RNA-seq and single cell RNA-seq, they are almost asking about, I want to know more about ChIP-seq, and my lab is doing the single cell ATAC-seq or doing some spatial transcriptomics. So I guess we'll keep the CGC team busy in the next few years probably. So in summary, so the workshop participants are a mixture of cancer researchers and tool developers, because this is a seminar in statistical department. So for cancer researchers, I feel like most of the benefit they get is easy access to data. So especially to large scale data, a lot of them are using the TCGA data and the GTEx data. But these are the HIPAA protected, and so they even don't have the place to store this data. And on the other hand, when they work with other members on the team, and they feel it is very easy to collaborate, you can simply add somebody else on the platform, and then they can look at the same data, same procedure at the same time. And so the large number of tools available at CGC is really, really helpful for them, as when they do different types of analysis, they feel like the tools are right there, and they can just click on it and put it into their pipeline. And especially, I think the thing that they like most is the interface is very, very friendly. Like a lot of times, when we teach a course, we ask them to use Linux or R, and they usually get lost in the middle. But this platform, the CGC platform, is so friendly that they don't have any issues with that at all. And in the meantime, so it's kind of surprisingly for the tool developers and more the quantity of people in the group, they were thinking about, when they develop the algorithms or the tools, they can actually take advantage of the existing tools at CGC and incorporate into the pipeline they are developing. And they also feel like it's a very nice way to share the tools they develop with a much broader audience. So in the future, we are thinking about, we have all the slides or videos available online. So this can be used as resources for other institutions or university if they are interested in learning more about the CGC platform. And it can also be used as great training resources for the next generation of cancer researchers. So this will end the first part of my presentation. So for the second part is actually as part of the discussion from the first part for the two developers, as the way they're thinking about develop tools and implement them onto the CGC platform. So this is a collaborative project with the CGC team for a specific package called SIGNET. So this package is essentially used for to construct networks, which is very computationally intensive because it use all the genes in the RNA-seq data and also all the polymorphism from the DNA sequence data. So here is the outline of the second part of my presentation. This project is led by my collaborator, Dr. Zhang, and this picture shows the overview of the SIGNET package. So from the upper left corner, so the package takes two types of the omics data. So one is the RNA-seq, which is transcriptomics data, and the other one is the DNA sequence, which we use the polymorphism or the SNP data as the instrument variable to infer the relationship or the directions between the genes. And part B is the pre-processing procedure. So basically the QC for both the RNA-seq and also the DNA-seq data. And once after the pre-processing, we can do what we call the cis-EQTL analysis to identify the instrument variable in the structural equation model to prepare for the network construction. And so the part D basically put all things together and build a model for each gene. And then we get the network as showing F. Once we compare, put them all together, so the last one, the part H, is essentially the final product. So you can see the computation is very intensive. It's not only from the pre-processing and also the EQTL analysis, but if you look at the part E, so it's kind of small, but it's a bootstrap data, which means to evaluate or to give an idea on how confident we are about the gene regulation. So we need to bootstrap the data many times. So usually is 1,000 or 10,000. So each one of them include maybe 10,000 genes. So it's very it costs a lot of computing time. And so here's another more toward the results from the SIGNET. So we can see that we use a bootstrap frequency to look at the confidence, and the part C shows the different networks that are constructed, and D is just a zoom out of one of the network. And after the network is constructed, so we can do some enrichment analysis showing E and F to see what are the biological processes and pathways enriched in the data that we are looking at. So there are many advantages of this algorithm. So first of all, in terms of the biological side, so this is a purely like data-driven gene regulatory network, and also is constructed on a genome-wide scale. So in this case, we kind of reduce the bias of the possibility of selecting any genes to construct the network, because here we basically use all the genes from the RNA-seq. And on the other side, so this algorithm can be easily extended to what we call the differential gene regulatory network. So essentially, when we work with cancer patients, so a lot of times we want to see what are the differences between healthy control and cancer patients. So we can actually build a differential network by taking both sets of data together in one analysis and get the differential network. Also, it will also show where the regulation is shared by both healthy control and cancer patients, and where the differences between these two groups. And computationally, I mean, we can take the advantage of the HPC, and more importantly, the cloud computing to make it much faster. On the right-hand side is some nice properties in terms of statistical theory and modeling. So we prove the oracle properties, and also the errors are bounded. So which means if we have large enough sample size, we always get the correct connection and regulations. And this is all based on the structural equation model and kind of connected to the Mendelian randomization that people use in practice. So here is the kind of procedure or four-step procedure for the SIGNET. So we start with the pre-processing, like the QC and data normalization, and then we can find the instrument variables that can be used to infer the direction of the gene regulation, and followed by the construction of network. The last one is to visualize them. So on the right-hand side of the panel is an example that we used in the application of the method. So this data is from yeast. So essentially, we take all the gene expression data and the SNP data and construct the network. So the shaded areas actually have certain biological meaning, which our collaborators, get a nice description of how these connections can be explained biologically. So more details, you can go to the scientific reports we published about 3 years ago. So in terms of with the CGC platform, it can actually make the SIGNET much more useful and easily to run the analysis. So the right-hand side shows the logo of the SIGNET on CGC. So I'm not sure if you can read it, but it says, "Welcome to the SIGNET." So the left is the bullet point. So essentially, the CGC provides a user-friendly interface of SIGNET, because originally we can only run SIGNET on Linux-based machine, which means it basically only people who know Linux very well will be able to use the algorithm, which, I mean, a lot of cancer researchers certainly won't be able to use it. And so secondly, it provides a very easily accessible workspace. So during our big data workshop in the summer, so we always help participants get lost in the analysis, even though we provide all the details. But somewhere in the middle, if there is something went wrong, they can never. I mean, it's really hard to fix them. But with the CGC, we don't need to worry about that anymore. And it's quite easy for them to select the tools and run the analysis. And the other thing is the reproducible workflow. So essentially, when we run the analysis, it's very common that certain software, if you have different versions, you always have this discrepancy, won't get the same results. But with the CGC platform, things, the pipeline is well built, and you can select exactly which one you're looking at, and we don't have any issue for the reproducibility anymore. And the last two is more regarding the customization, the downstream analysis. So because there is a long procedure with many steps. And so for some parameters in the middle, so if a certain researcher just interested in a particular chromosome, a particular region of the chromosome, you can easily change this with the CGC platform, but previously we have to modify the code to make this happen. And finally, the cloud computing provides a very nice way that we can analyze large data sets. We can analyze a lot of them simultaneously. Otherwise, it won't be almost impossible. Okay, so here is an example about about the platform in terms of the testing. So we before we run the real TCGA data, so we just simulate the data based on the TCGA to test the package. So the Seven Bridges command was used to upload the data from our local server to the CGC. And here is just a very one line of command, and you can see that we have the all the things listed nicely after this. And the other thing is, I mean, for all the bioinformatic packages, so we always have the issue of the computing environment. So to avoid the issue, one of our PhD students, Zhang, actually developed a Docker image for SIGNET and uploaded to CGC, and then we don't need to worry about installing any packages or anything like that. So here is the procedure of the pre-processing of the RNA-seq data. So the top panel is a description of the procedure. It gives the introduction how this was done, and the bottom panel shows the pros of the ID label and also different characteristics associated with the input files and also the settings with the output. So it just makes so much easier for the to reproduce the analysis. And so this is the user interface for the tool. And the fonts are probably too small, so you may not be able to read it. But yeah, it's fine, just to show the idea about how nice the user interface is. So on the left part, the user can modify the input files by just click on the arrows or the question marks. And in the middle, so you can change the settings. So this is the one that I just mentioned. So if you don't want to do the entire, like all the chromosomes, you can specify the chromosomes you want to work with, or you can restrict to even regions. So it's quite easy to put restrict the settings here using the app settings. And on the upper right corner, there is a link on wheel stats and logs. So this one, you can look at all the intermediate steps and all the logs. And if you want to change the settings and rerun the analysis, you can just click on rerun. So it's very user-friendly and convenient to change anything in the middle or after it's done. So here is the example of the running logs for the SIGNET. So in the on the left panel, we get all the logs during the analysis, and the right panel shows the work in progress record using the SIGNET. So here is example about just looking or exploration of the result. So it's just a bunch of numbers, but so essentially you can use the bar on the top to look at any part of the results during the process, and you can also copy the results, download it, or whatever you want to do. And this one is basically the second step. Like after the pre-processing, we do the EQTL analysis to find the instrument variable. So the description box describes the procedure, how we identify the cis-EQTL as an instrument variable. And so this is in terms of both the common variance and also the rare variance, and we give the options, for different things you want to do. And at the bottom is a pros again with all the ID type and other things that related to this analysis. And here is the results of the output from the analysis. So again, the left panel contains all the logs with the output files. and the right panel is the procedure of the analysis. So this will provide the analysis both on the common SNPs with high minority of frequency and also the rare variants, which we use the burden test to combine the SNPs for the eQTL analysis. And so this one, it shows the power of the CGC. So since in the cis-eQTL analysis, we actually looking at all the genes and using all the SNPs from the genome-wide scale. And so it's very time consuming. So that's why if you compare, the time used with different instances, so the left side panel is a different instance you can select. And so we basically select 2 of them for comparison. The middle one is based on using 2 CPUs, and the one on the right is based on using 8 CPUs. So you can see the time, like the bottom one with 2 CPU took about 6 minutes, and on the when it changed to 8 CPU is less than 3 minutes. So that's where the power of the CGC coming from, and this is only for a small kind of simulated data based on TCGA. So our goal here is to, when we analyze the real TCGA data, so with the appropriate selection of the instances, so we expect the computation time will be much more efficient than using other regular HPCs. So this is still a work in progress, and we got tremendous support from the CGC team, and the original development algorithm was helped by our senior IT specialist, Dr. Abel, a lot. But once we started to put on CGC, so we are frequently visit their office hours. It's actually on a weekly basis. And sometimes, since we have a large group, we couldn't make the same time, and they're always willing to schedule a separate time to meet with us and solve the problem. So that's why, we started the project actually after the workshop. It's kind of a couple of months ago, but it progressed really well. And so we owe a lot of thanks to Zhang, David, and Thea for their immediate help. Whenever we have some problem, they almost solve it, help us solve it, quickly. So in summary, for the second part, as an example of implement newly developed algorithms onto the CGC platform, so it can make the package much more user-friendly, and we can easily use the data that are already on CGC to do the analysis rather than uploading, do all the KRC, especially kind of challenges with the large data set. So again, it's easy to collaborate with large groups. We can have we all have access to to the same project, and obvious computation is very, very efficient. So in the future, we are thinking about, so once we finish the testing, we will analyze all the different cancer types with the TCGA and with some other data sets. So this will help us, create, I would say, a knowledge database for all the regular network for all types of cancers or subtype of cancers. And also combined with the GTEx data, we can come up with a differential network between the cancer tissue and healthy tissue from the GTEx. And so I'm only the spoken person here, so all this work has been done. It's kind of years of work for graduate students, postdocs, and collaborators. So we also have, acknowledged the data resources from the dbGaP in terms of TCGA, GTEx, and the funding coming from different programs of NIH. Mostly from NCI, but also some of the methodology development from NIA and NIGMS. So with that, we also owe a lot of thanks to the CGC team in terms of the both training workshops that are funded by NCI and also the algorithm development as part of the research effort. Okay, with that, I would like to take this opportunity to advertise our upcoming workshop on big data next summer. So it's May 19 to 28th, and this is the first time we have it in person, because the first year of the project was 2020, and we got everything ready for in person, but then the COVID started. So we are excited to kind of welcome everybody to come to Purdue campus and have the in-person experience with the workshop. And I will stop here and take any questions you may have. Yeah, thank you all for your attention.
Thank you so much, Min. This was great. It's been great collaborating with you. We have a question for the first part of the talk from Sam Payne. For the first series of the lectures, how much time was the lecture and how much time was it to solve the problem?
I would say, because it's ongoing, like everybody logged in at the beginning of the lecture, so we don't have a clear cut of the split, because, so maybe if the brief introduction, I would say 10 to 20 minutes. Or maybe, you're probably the best person. How do you remember like the time you spent just by talking, as everybody started to log in and get really excited or try different things?
Yeah, so from the initial, for the first lecture, it was mostly just a presentation, and the last 5 to 10 minutes were the students logging in and creating their first project. The second lecture, so the fourth lectures were each about an hour. The second lecture was mostly like 10 minutes. Phil just presenting what the workflow was going to be, what kind of analysis we were going to do, and then the students would log in with him, and he would stop at every point and ask if people were done. I do recall a couple of times people getting sort of lost because they needed to go into our studio, but we helped them go through solve through their issues using the chat function, or we basically waited for the person to be done so that we can all move on. Yeah, so the third lecture was a little bit less. So third lecture was also bulk RNA-seq. The fourth lecture for single cell RNA-seq was a little bit less hands-on. There was more explanation of what single cell is and how it was different from the bulk RNA-seq, because we had given that background in the previous lectures. But the students did log in at the end and ran their own Seurat workflow using Data Studio.
Yeah, thank you. Yeah, but I do remember things like you guys actually provide the material before the lecture, and a lot of, at least the students started to, they couldn't wait. They started to try already before the lecture.
Yeah, so we did provide some materials in advance, so I think that might have helped.
Yeah, I think that's really nice. I think because some of the students, they were trying to use this as in their research projects, and they just couldn't wait and keep asking me, "Oh, when do we get this?" And when you guys provided that, and they said, "Oh, now I tried this, so they started to ask questions already."
Nice. Yeah, I do remember that, and I do remember a couple of your grad students coming into office hours for months after trying to get, for things that they weren't working on themselves. They were still following through and doing their research project using our classes. So yeah, it was nice to see that they were continuing using the CGC after our workshop.
Can I follow up on that?
Absolutely.
You had URLs posted to videos. Is it possible to put those in the chat so that we can get those?
Yes, I can post them in the chat, but at the end, so at the end of the webinar, I will post the slides on the cancergenomicscloud.org, and you'll be able to download the slide deck and click on them. But yes, I think we can put them on the chat as well, if that's easier.
Thank you.
Of course. And do we have any more questions for Min? Okay. my usual question for Min, you already answered, which is how has the CGC helped you and your students? But you already answered through that during your talk. So if people don't have any more questions, we can adjourn. Rowan?
Yeah, I'll ask a question. So I got really excited about this next year Purdue workshop that you are going to be doing in May. Do you have any idea of what type of topics that we would want to cover for the students? I'm getting ahead of myself, obviously. It's like in several months, but it's exciting to me, and I want to make sure that we have the relevant materials. So if there's any particular thing that you think the students are interested in besides single cell RNA or anything else, I'd love to hear about it.
Yeah, that's a great question. So we always do like post workshop survey, after we did, I think last year, because the way it works is we wrote the proposal, and so with 4 topics right now. So it's the bulk and single RNA-seq and chip-seq and GWAS and network analysis. And we couldn't change it for our workshop, but we can plan for the future. And also for you guys will be much more flexible, because this will be kind of an add-on, for complementary information. And I think, one of the popular ones is the ATAC-seq and the single cell ATAC-seq. And a lot of them are starting to ask about spatial transcriptomics data, which is pretty new. I don't know how many pipelines are there already, but that's one of the 2 major ones that people keep asking. And also I would say the microbiome data analysis is keep coming up. And people also talk about the proteomics, metabolomics, those kind of things. So those are the things we touched a little bit just by giving, like, you invite somebody to give a special lecture, but never kind of put it into the hands-on session for those kind of data analysis. Yeah, so if you guys could describe some of those, that would be really helpful.
Yeah, we have time to work on some of this stuff and make sure it's available publicly so that everyone can follow along.
Yeah,
That sounds
definitely.
good. I'm excited about it.
Yeah. And also we will be able to see each other on campus, You're helping
I think that
online.
will make it a lot easier to teach. I love teaching people and helping with things. There are limitations virtually. You can't really see the expression on people's faces. So being there in person, I think will be a lot easier to have people follow along, so we can give them individualized attention if needed.
Yeah, definitely. Yeah, basically all of us stay in the same room from 7 a.m. to 10 p.m. with just even have lunch. We have lunch at the same place in the across the hallway of the computer lab. We only go out for dinner, like a 1.5 hour break for dinner, and that's it. And we stay together for 10 days. We really build like strong relationship, between all the participants, and some of them are still kind of collaborating even after the workshop, because they do a project together. And then I think the reason we have to stop at 10, because that's the building deputy. They came and locked the door of the computer lab, and then they went back to the dorm, and there's always computers in the obviously they keep working together, and until they finish or something, it's like, oh, we have been together for so long. We just end up doing build up long-term relationship in the future.
Well, that's a great way to establish collaborations. So I'm glad to hear it.
Yes, can't wait to be part of that this year. Be excited.
So, Min, how many of your attendees are local Purdue people versus other people coming to Indiana?
That's actually why we have the CGC come here. We usually have 2 out of 30, because the idea is to really have recruit people nationwide, and so that's why most people actually, I mean, Midwest probably have a little bit more representation, just because they don't have many resources as both coasts. But Purdue people, I think is either 1 or 2, never more than 3.
So it's not just all of your chemistry grad students coming and figuring out
no, they're actually all. Yeah, we originally sent the flyers to all the NCI designated cancer centers. So, and then almost always it's people from one of the, I think 79, the NCI designated cancer centers. But then later on we realized, there are also other cancer centers out there besides the designated cancer centers. So we started to send them to, kind of more institutions, beyond the cancer centers. Like if there are some department, they are working a lot on the cancer projects. So we also send to those departments too.
And they're mostly students?
No, we actually have quite a mixture of participants, from all the way from students to the dean of school medicine. So, because I think for the higher level people, I even asked them, why do you want to come to the workshop? They said, we need to give the money to somebody to support the research. I need to know what this is really about to make sure I'm not biased when distributing the funding. So
That's
it's
good to know.
Yeah, and they actually have, that helps the discussion a lot because, when, for people at different level of their career, so they have different perspective for the same thing. So whenever we talk about a certain topic, you always have different views, like from the grad student, junior faculty, senior faculty, and the admin people. So, and then it end up with a much longer discussion than we thought. So we're always behind the schedule, but that's okay. Since we stay over late into the night, we can always make it up. But it just, I think, having a mixture of populations is helped a lot with the discussions and topics, and especially when it comes to the group project, and then they will take care of different aspects of project. And I remember there was one particular data set that they even did analysis in a certain way that nobody else has thought about before, because they come from, like, there's a fellow, a grad student, and a senior person, based on many years of her research, thought, we should look at the other gene and how they are correlated. And then, they actually have some very interesting findings from that.
Cool. Awesome.
Yeah,
Thank
so
you.
it's the same data, but, when different people analyze it, the results are, kind of all over, like, that's actually make it very interesting toward the end of the end of the workshop.
Yeah, that's great. Thank you so much, Min. Thank you, Sam, for being super interactive. This was a great webinar. This will be the last of 2022. For this year, we will restart again January 2023 with a new series. And thank you all for coming, and thank you, Min, for your great presentation.
Okay, thank you.
Thanks.
Thank
Bye.
you.
Bye.